Hello everyone. Today we are going to continue the chapter number 5 molecular basis of inheritance, and this is theory part 11. So and this is in this we are going to cover the NCERT section 5.10. So basically in today's class we are going to understand majorly about DNA fingerprinting. Okay. So you guys have even seen many crime series or any series in which you have seen, okay, so we have to match the match, like is like we have to match the culprit, or we have to match the that the child is belong to that specific parents or not. So we will do the DNA fingerprinting, we will do the DNA test. So today we will understand the basis of this DNA test and this DNA fingerprinting. So let's get started. So here's the disclaimer. So what are going to be today's learning objective? First, it will be that we will understand that what is the basic principles behind this DNA fingerprinting. Then we will identify key steps in the DNA fingerprinting process, recognize application of this DNA fingerprinting in forensic, paternity testing and all. Then today's index is like this. We are going to cover NCERT section 5.10, which is DNA fingerprinting. Then there will be a summary slide that always is there, where we will going to discuss like what are the key points, not key points, what are the summary, that we will summarize the whole class for you in that. Then there will be some NCRT based question that we gonna solve together. Then followed by keynotes, and lastly there will be some interesting questions that you guys gonna solve yourself. So before starting with this fingerprinting, technically we are starting DNA fingerprinting NCRT section 5.10, but before actually understanding what are the different steps in fingerprinting, we have to understand some basic concepts behind it. So first list we have to understand that what are the different variations that are present in human genome. So if we talk about some key points, some variations that are present in human genome, first is the sequence similarity. So in yesterday class, we have seen that from this human genome project, we got to know that 99% of human DNA based sequence is identical across individual. Means the 99.9% of human DNA based sequence is similar in across all the individual, everyone. Then if we talk about the genome size is 3 into 10 to the power 9 base pairs. So now if you are saying that 99.9% of the sequence of the human DNA base sequence is similar, then let's calculate what are the base sequence which are different. So definitely of 100 minus 99.9% is what? 0.1% of genome varies between the individual, and if you will calculate 0.1% of 3 into 10 to the power 9 base pair, then it will come approximately 3 into 10 to the power 6 base pair sequence differ between individuals. Means this amount of base sequences are different among the individuals. This is giving what? This is giving the phenotypic uniqueness. These DNA sequence which are different, okay, these differences lead to unique physical trait, unique phenotype, and that's why we all look different. We all, do we all are different. So from this slide, what is the summary? So from this slide, you guys have understand that there is variation in human genome sequence. There is 99.9% of sequence similarity. Means the human DNA base sequence, 99% of the human DNA base sequence is identical, is similar across individual. Then if we talk about the size of the human genome, then it is 3 into 10 to the power 9 base pairs. Now if we talk about if 99.9% is sequence similarity, then what is the base sequence differences? It is only 0.1% of the genome which varies between the individual, and which is approximately how much base sequence? It is 3 into the 10 to the power 6 base sequence which are different, which differs between individual. And this difference, okay, This DNA sequence is giving unique physical trait, unique phenotype to the individual, and that's why we all look different. Okay, all have different phenotypes. Now, why there is need of DNA fingerprinting? Challenges of full sequencing. So, first of all, it is not possible to sequence the entire genome. You know, it is very time consuming and costly. So, it is very challenging. If you will compare, even it is very difficult to and sequence the entire genome to identify difference. If we are talking about that there is 0.1% of that which is different, so even if you calculate that, so comparing these two set of 3 into 10 to power 6 base pair is also very difficult. So what was the solution? The solution is DNA fingerprinting, as it is a faster method for comparing DNA sequences between individuals. So I hope you guys understand what is the difference between. So now let's come to repetitive DNA. Now we have to understand what is a repetitive DNA. So this is these things we are studying because we are making the basis of DNA fingerprinting. So what is the repeated DNA? So these are the short stretches of DNA. Okay. And in this region, okay, in these short stretches of DNA, the DNA sequences is repeated multiple times. Okay, that is the repetitive DNA. Now we will talk about satellite DNA. This is NCERT. From here, you guys can answer NCERT exercise 8A. So now, so when the density gradient centrifugation has been done for this repetitive DNA, density gradient centrifugation, you guys understand, when we centrifuge and then on the basis of the density, the things got separate. This is what we call density gradient centrifugation. So this density gradient centrifugation separates repeated DNA from the bulk genomic DNA, forming smaller peaks known as satellite DNA. So when the density gradient centrifugation has been done for the bulk genomic DNA, we understood that what happened, this repeated DNA got separated on the basis of density from this bulk genomic DNA, and they termed it as satellite DNA. It is forming a smaller peaks, and that's why we termed it as satellite DNA. So here you guys can see, like, after this density what happened, this is the centrifugation that has been done, this like it came, and there you guys can see there is like two peak. Okay, first is the main band and then there is this small peak, then this is what we call satellite band, and this is what like peak of satellite DNA. It was discovered as a small satellite band during density gradient centrifugation and named as satellite DNA. So what happened when this came, when this density graph came, so this small peak, it was like it was discovered and it was named as satellite DNA. So what we understood from this, like this slide, that what is a repeated DNA? Repeated DNA are the short stretches of DNA, and DNA sequence in that region is like repeated multiple times. And when the density gradient centrifugation has been done for the bulk genomic DNA, so this repeated DNA separate out, it gave a smaller peak, and this smaller peak is discovered as satellite DNA. So this is the whole everything in this slide. Now this satellite DNA is categorized based on, so we have categorized this satellite DNA on basis of what? Base composition, like whether it is AT rich or GC rich, its segment length, and the number of repetitive unit. More the number, it is microsatellite. So less the number, it is microsatellite. More than that, it is mini satellite. So this satellite DNA is characterized on which basis? First, base composition basis, which is AT, whether it is AT rich or or GC rich. Then what is its segment length, and then number of the repetitive units. Is it micro or is it mini? Now, what are the characteristics of what of this satellite DNA? Non-coding region. So, these repeated sequence do not code for proteins. Okay, typically do not code for protein, but it constitute a significant portion of the human genome. Because in yesterday class, when we were talking about HGP, we have studied what, like only less than 0.2% of whole genome codes for protein. So you guys can think how low amount of it's going to code for protein. So that's where, and this is also like linking here that this repeated sequence, they are like constituting a significant proportion of human genome, but they do not code for protein. Second, polymorphism. So now see, from here, from this polymorphism, now you guys listen properly, because this is what going to make the basis of DNA fingerprinting. So polymorphism, what is this polymorphism? Repeated DNA sequence exhibit high degree of polymorphism. So this sequence, this repeated sequence which we have talked about, like like how this satellite band came and all those things. So this repeated sequence, what is happening? So this repeated sequence exhibit a high degree of polymorphism. Means they have a variation among the individual. Okay, so they have seen the okay. These are repetitive DNA sequence. They are not even coding for proteins. Okay, but they are constituting a significant proportion of human genome, and it is showing a high degree of polymorphism. Means it is showing a high degree of variation among individuals. Okay, among individuals in the population. Now, this polymorphism is the foundation of DNA fingerprinting. This polymorphism became the building block, became the foundation of this DNA fingerprinting. So, what we have studied in this slide, that what are the characteristics of this satellite DNA? So, the characteristic of this satellite DNA is that it is non-coding, means it is not going to code for proteins. It constitutes a large proportion of human genome, and this repetitive DNA sequence, it exhibit a high degree of variation of polymorphism, and this polymorphism became the foundation of DNA fingerprinting. So till now what we have studied in today's class, so till now we have studied what is the like variation in the human genome. 99.9% were identical among individuals, then 0.1% is variable, is is different. When we have calculated to, it came out to be 3 into 10 to power 6 base pairs. But it is not possible when we are comparing the individual's genome, it is not possible to even sequence this amount of genome. So then what came? Then this DNA fingerprinting came into the picture. Then we were like, okay, so this is the faster method. Okay. Then what happened? Then we have studied about this repetitive DNA, the small stretch of DNA in which the sequence is repeating again and again. So when this what happened when this density gradient centrifugation has been done from whole genome, they have seen this repeated sequence got separated and it is giving a peak, what the peak we call satellite DNA. Then what happened? Then now we are studying the characteristic of the satellite DNA. And this is the non-coding region, means these are the repetitive sequence which is not going to code for what, which is not going to code for protein, and it constitute a huge amount of human genome. And the basic, the the what is the basis, what is the foundation of fingerprinting came out to be from this is that this repetitive DNA sequence, it exhibit a high degree of polymorphism, means they are showing variation in individuals among population. So this is a thing. Now, this DNA polymorphism became the basis of DNA fingerprinting. Let's talk about its definition. Polymorphism refers to variation at the genetic level within a population. This is from here. You guys can answer NCERT exercise 14b. Now, what this polymorphism refers to, as I said, it is a variation at what level? At the genetic level within a population. It is the foundation for genetic mapping of human genome and DNA fingerprinting. Polymorphism range from single nucleotide changes to large scale genomic alteration. DNA polymorphism is traditionally defined as allelic sequence variation. More than one variant, that is allele, exists at a specific location, what we call locus, in the human population. If a heritable mutation is observed in a population at a frequency greater than 1%, it is considered as DNA polymorphism. I understood that this is like quite confusing. Let's answer you the let me understand you this concept in more basic terms. So what it is saying, it is saying that what is like what is the polymorphism? Polymorphism is what? It is the variation at what level? At the genetic level, okay, at the genes level among the individual within a population only. Okay, it helps in genetic mapping, means scientists use these variation to find where genes are located on chromosome. Okay, so these variations are helping scientists to find where genes are located on chromosome. These difference can be small, can be as small as like single nucleotide change, like one letter change. Example, A got replaced by G, means adenine got replaced by guanine. Or even this can be large scale genomic alteration, like even like missing extra DNA pieces. Okay. Any the whole DNA pieces got missed. Okay. So these can difference can be as big as possible and as small as possible. Now, from what fourth point is saying, let's understand. This polymorphism usually involves what? This polymorphism usually involves alleles. Okay, one gene can have multiple forms, forms, what it is, alleles in the population. So this usually involves what? This usually involves allele sequence, like gene, like how to tell you, like one gene can have multiple forms, multiple alleles in that population only. Okay, it is like that. So DNA polymorphism is what? It is a traditionally defined as allelic sequence variation. Because why? Because we know that one gene can have multiple forms. Multiple forms means different alleles in the population. Then it happens at a specific position on the DNA. This is what we call locus. We know that these allele exist at specific location, what we call locus. The same gene location can have different version in different people, because now we say this is the allelic sequence variation. So at the same position, you know at the same position, at the same locus, same gene location, they can have different version in different people, because we know there is variation in this allele exists. So at the same position, at the same locus, for the same gene location, they can have different version in different people. Okay, because we know that one gene can have multiple forms, alleles in the population. If a population, if a mutation, okay, if this mutation, if this change is passed on and seen in more than 1% of the people, it is called DNA polymorphism. Understood? What is this? This is like first we have telling that what is the polymorphism? It is the variation at genetic level within a population. Okay, it helping scientists in genetic mapping. How? Because these scientists use these variations to find where genes are located on chromosome. Then these differences can be as big and as small, like one letter change can be there also, or even there is like big piece of DNA that is missing. This involves what? This is like important point to understand. This usually involves alleles, okay, gene versions. One allele, one gene, sorry, can have multiple forms, alleles in the population. Okay, and we know that this happens at a specific location on the DNA, what we call locus. So the same gene location can have different version and in different people. And if this mutation, if this change is passed on, means it is herited and seen in more than 1% of people, it is what we call DNA polymorphism. So now let's understand how it has been used. Now, if we talk about this origin of polymorphism, so we have talked about the basic points of this polymorphism. Now understand what is the origin of polymorphism, and it may create like more clarity on this topic. That polymorphism arises due to mutation in what? In DNA sequence. Mutations can occur even in somatic cells or germ cells. The probability of observing variation in non-coding DNA sequence is always higher. Mutation in non-coding regions are less likely to affect any reproductive ability. These mutations accumulate over generations, contributing to this variability and polymorphism. So, we have talked about this polymorphism, like how it, like what is gene version. It can be exist in multiple forms, means alleles at the same locus, and that's why we call it as polymorphism and all those things. And so now, what is the origin of this polymorphism? This polymorphism usually arises due to mutations in DNA sequence. This mutation can occur either in somatic cell or germ cell, and this probability of observing variation is mostly in non-coding DNA sequence. Okay, it's not in the coding DNA sequence. And because these mutations are in non-coding sequence in regions, so this is going to less likely to affect any reproductive ability. These mutations accumulate over generations, and this is what it is going to, like, this is how it is going to contribute to polymorphism, or what we call variability. Okay, now we will move to DNA fingerprinting. So now how this, they are using this polymorphism for DNA fingerprinting. Now we will understand. DNA fingerprinting relies on identifying difference in specific region of DNA called repetitive DNA. From here you guys can answer NCERT exercise 13. So, what this DNA fingerprinting relies on? So this DNA fingerprinting relies on, as we told in previous slides, that polymorphism is the foundation of DNA fingerprinting. So this fingerprinting, DNA fingerprinting, it relies on identifying the differences, identifying that polymorphism in specific region of DNA, what we call repetitive DNA. And we have studied out what is repetitive DNA in the previous slides. It was developed by Ellis Jeffrey. What was the principle behind this DNA fingerprinting? So the principle behind the DNA fingerprinting is that the LF Jeffrey utilized this satellite DNA with high polymorphism as probe. This highly polymorphic DNA probes are called variable number of tandem repeats, that is VNTRs. So what this fingerprinting relies on? So this fingerprinting relies on identifying the differences in this specific region of DNA, what we call, on which specific region of DNA? Repetitive DNA. It was developed by Alec Jeffries. Now, what was the principle behind this? So here you understand later, more you will understand in upcoming slides, that LF Jeffrey utilized this satellite DNA, the satellite DNA which we have talked about. So when we, density gradient centrifugation was done for whole genome. So what happened? This repetitive DNA separated and gave a small peak, what we call satellite DNA and all those things. These also having what? Repetitive sequences. So this scientists utilize this satellite DNA who has high polymorphism, means who has high variation in the individuals among the population as probe. Probe, you guys remember what we have studied in before biotechnology chapter, that what is probe? So probe is the small stretch of what DNA or RNA that is complementary to the specific part of the DNA. So what the scientists did, they utilized this satellite DNA who has this high polymorphism as probe. And this highly polymorphic DNA probes are called what the name has given to this DNA probe, which is polymorphic in nature, variable number of tandem repeats, VNTRs. What? Variable number of tandem repeats. Now, what is this variable number of tandem repeats? It belongs to a class of satellite DNA referred to as mini satellite. VNTRs are short DNA sequence arranged tandemly in many copy numbers. Copy numbers varies between chromosome within an individual. The number of repeats show very high degree of polymorphism. The size of this VNTR varies in size from zero point 0.1 to 20 kb due to this polymorphism. So understand now. We have told that the Alex Jeffreys, what he did, he used this satellite DNA with high polymorphism probe, and he named that probe, he has given the name that probe what? Variable number of tandem repeats. So now we are understanding what is this variable number of tandem repeats. This belong to satellite class of a satellite DNA, that is mini satellite. Now, what is this VNTRs? They are the short DNA sequence. Okay, short DNA sequence, and they are arranged tandemly in many copy numbers. So here in this figure you guys can see what is this? This is GATA we will say here. GATA, okay, this DNA segment. Now you guys can see this GATA has been repeated 4 times here. Okay, 4 repeats. And in next is what? It is repeated 6 times. So this is how this short tandem repeats look. So VNTRs what? These are the short DNA sequences which is arranged tandemly in many copy numbers. Like for this it is 4, for this it is 6, for other it might be 8, 10 or whatever. Now, this copy number, as I told now, this repetitive, this tandem repeats, these copy numbers, like whether it is 4, 6, 8, 10, 15, whatever, that copy numbers varies between chromosome within an individual. So these copy numbers varies between chromosome within an individual. So this number of repeat show very high degree of polymorphism. So this number of repeat will show like how much this degree of polymorphism is. Now the size of this VNTR varies in size. It can be from zero point 0.1 to 20 kb kilobase pairs due to this polymorphism. So you understand like how much variation it does have, as this VNTR can differ in size from zero point 0.1 kilobase pairs to 20 kilobase pairs. And this, okay, so this is what the variable number of tandem repeats. Now comes the methodology of DNA fingerprinting, like how this DNA fingerprinting has been done. Southern blot hybridization using radio-labeled VNTR as probe was used earlier for DNA fingerprinting. This technique includes following steps: DNA isolation, restriction, digestion, electrophoresis, blotting, hybridization, autoradiography. So these are the different steps. Okay, these are the different steps performed in this technique, this DNA fingerprinting technique, which has been, these steps have been used. So we will read about each and every step properly. Okay, so let's start with that. Okay, before that, I want to tell you like what we have studied, what we have seen here. Sudden blot hybridization using radio labeled VNTR as a probe was used earlier for DNA fingerprinting. What does this mean? So first of all, you have to understand this sudden blot hybridization, what it is, sudden blotting, what it is. It is a molecular biology technique and it is used to identify specific DNA sequence within a DNA sample. Okay, so this is nothing, it is just a technique, it's a molecular biological technique which is used to identify the specific DNA sequence. And how we are doing this, like how we are identifying this specific DNA sequence? So we are identifying this specific DNA sequence using radio label, you understand? Okay, and using radio label VNTR, okay, variable number of tandem repeats, which we have studied in previous slide, as probe was used for earlier DNA fingerprinting, and the steps which are involved in this are given below, okay, given in front of you guys. Now I will make you understand each and every step. So first step is the DNA isolation. Definitely, we have to identify a particular DNA sequence. So now we have to get what? To identify the DNA sequence, to the specific DNA sequence, we need the DNA sample. So first we will do DNA isolation. So when we will do the DNA isolation, what will happen? Genomic DNA is extracted and purified from the sample. First we will what we will do? We will extract this genomic DNA, and then we will purify it. Then using restriction digestion, we know what is restriction digestion. Like we use this restriction enzyme which can cut the DNA at a particular time, at a particular place. So first we have isolated the DNA. Then what we did? Then we use the restriction digestion. We use this restriction endonucleases to cut the DNA into fragments. So this is the whole DNA fingerprinting schematic representation of the DNA fingerprinting that has been given in NCERT, so that you guys will understand. I will make you understand at last when we will study the all the points. So first we have studied like what is the first step? DNA isolation, in which what we did, we have extracted the genomic DNA and purify it. Then what we did, we using the restriction endonucleases, we have cut this DNA into fragments. Now, third is the electrophoresis, means what we did in this step. So, in this step, we have separated the DNA fragment based on their size. Now, we did this plotting, this sudden plotting. So, what we did, the separated DNA fragment, what we got from this electrophoresis, we transferred it from the gel onto what? Onto a synthetic membrane. What that synthetic membrane is made up of? Nitrocellulose or nylon. So these are the what step? First we did DNA isolation and purification. Then we make the fragments of the DNA by using restriction endonucleases. Third, using electrophoresis, we have separated the DNA fragments on the basis of the size. Then we did this blotting step, and what we do in this blotting step, we that DNA fragments that we got from the gel electrophoresis, we transfer it from gel onto synthetic membrane, and that synthetic membrane is made up of either nitrocellulose or nylon. Then what we do? Then we will do the hybridization. Now the membrane is incubated with the radio label VNTR, that is variable number tandem repeats probe. So that membrane on which we have now on which we have keep that on which we have plated our this DNA fragments from gel to that nitrocellulose membrane. That membrane has already what? Has already radio label VNTR, variable number tandem repeat, that we have studied, that probe. Now what will happen? This probe is going to bind to its complementary DNA sequence, and that's how we will identify the DNA sequence. So now you guys can understand what happened in this step. This is what? This is a schematic presentation of DNA fingerprinting. Okay. Few representative chromosomes have been shown to contain different copy number of VNTRs. For the sake of understanding, different color schemes have been used to trace the origin of each band in the gel. The two alleles, paternal and maternal of a chromosome, also contain different copy number of VNTR. It is clear that the banding pattern of DNA from crime scene matches with individual B and not with A. So what is happening in this figure? Let's see what is happening in this figure. So this is the maternal chromosome and this is the paternal chromosome. You guys can see even in this maternal and paternal chromosome, this copy number is so differing so much. Okay, you guys can see they contain different copy number of VNTR. In this you can see only 3, in this you can see 4, and many on this. Okay, now what happened? First they have did what? First they have did this. First they have did this DNA isolation, then purified it, used restriction endonucleases and all those things. Then they have run this gel. So from when they are running this gel, you guys can see that what is it? Smaller fragments are coming behind and coming beneath, and larger fragments are above that. So they did this first. So this amplified repeat separated by size on a gel gave a DNA fingerprint. Okay, this is the DNA from crime scene. Here you guys can see this is the this DNA fragment what you have seen on the gel is from the crime scene, and this is what this is from the individual A and individual B. And you guys can see what is happening. It is there. It is very clear that the banding pattern of DNA from the crime scene, okay, matches with individual B and not A. So here you guys can see that this scene from crime, this gel from the crime scene, okay, this crime scene and this individual B, they have like kind of equal number of short tandem repeats. Like you guys can see if this red is matching, this red is matching, this yellow matching, whereas this A don't have anything. Okay, so this is from individual A, this is from individual B, and this is from the crime scene. So here we guys can see that B is matching with the crime scene. Now, what happened? Hybridization. Currently, many different probes are used to generate DNA fingerprints, that is, oligonucleotide probes, cRNA, and PCR amplified DNA sequence. So, nowadays, a lot of so you nowadays you can they are using many different probes to generate this DNA fingerprints. For example, this oligonucleotide probes, cRNA probes, what we call ribo probes, PCR amplified DNA sequence, many more. But we have studied that these things are VNTRs that are used mostly earlier. What they do after that, after hybridization, the hybridized DNA fragments are detecting by exposing the membrane to what? To X-ray ray. This reveal the location of VNTR sequence. So this like how we see it, how we visualize this. We use the autoradiography in which this hybridized DNA fragment, okay, are detected by exposing the membrane to X-ray film, and this reveal the location of VNTR sequence. Autography gives what? Autoradiography gives many band of different size. These bands give a characteristic pattern for an individual DNA. So these are what? These are the characteristic pattern for an individual DNA. Okay. So here we can see that how this, crime scene persons is matching with this individual B. Now, if we talk about what are the different applications of this DNA fingerprinting from like this, you guys can from here you guys can answer the NCERT exercise 13. So, crime investigation, it helps in identify the criminals. That the same example which we have given in the figure, that how the crime scenes is matching with the individual B. Then connecting suspect to the crime scenes. So this is the identification of what? Criminals by DNA fingerprinting. What they did, they collected the DNA, they collect the crime scene evidence, and then there was two suspects. Okay. So they perform the PCR on repeats. When they have seen on this crime scene, there was in one chromosome it was 12 repeats, and on another it is 16 repeats. And from suspect A, they have shown for like first one chromosome, he has also 12 chromosome, 12 repeats, and on second, uh, he has 12 repeats, whereas suspect B has same, that is first he has 12 repeats and then he has 16 repeats. So what is happening when they did the gel electrophoresis, so you guys can see this DNA crime scene evidence is matching with the suspect B. Okay, so this is the one of the like this like bands you can see clearly, and it is better to understand. Then family matters, it helps to prove parentage or testing for of paternity. It helps in resolving family dispute and immigration cases. So here you guys can see This is the DNA fingerprint profile of child and parents. Okay, so this is the fathers, this is the child, and this is the mothers. And then you guys they can like give that what percentage of similarity was there and all those things. And thus it is used to solve these paternity cases and all. Then the scientific research, they can use this to study the genetic diversity, understand the human evolution. Then can be helped in medical research and all. So, what was the summary of today's class? It is that DNA fingerprinting is a technique used to identify individual by analyzing what? By analyzing their unique DNA pattern. It examines specific region of DNA that vary between people but are identical in all cell of the same person. So here you guys can see what is like main difference, that it examines the specific region of the DNA that vary between the people, okay, vary between the individuals among population but are identical in all cell of the same person. The process involves what? Extracting DNA, cutting it with the endonuclease enzyme, then separating the fragments through electrophoresis, and then creating a distinct banding pattern. This method is widely used in forensic science, paternity testing, and identify victims in mass disasters. Okay, this identify victims in the mass disaster, like for example, like like previous month that plane crash happened, Ahmedabad plane crash. So body was so like it was burned so badly that it was very difficult to identify the victims. So at that time they used DNA fingerprinting, and this DNA fingerprinting, so like people can understand or got to know ki, okay, this is from our family and all, and then they can take the body. So yeah. It's highly accurate because no individual except identical twins share the same DNA pattern. So, this method is very accurate. Why? Because except the identical twins, okay, except this identical twins, there was no one who's going to, share this same DNA pattern. The identical twins only share the same DNA pattern. So, that's why it is very highly accurate. Now, let's solve some NCERT based questions so that we can get more clarity on this chapter and on this position on this like portion. Who developed DNA fingerprinting? James Watson, Francis Crick, Sir Alec Jeffrey, or Rosalind Franklin? So it was Alec Jeffrey who like developed this DNA fingerprinting. He utilizes what? He utilizes the VNTR's probe, that is variable number of tandem repeats probe, to detect the DNA bands by southern blotting technique. Watson and Crick did what? Watson and Crick discovered the DNA structure, not the DNA fingerprinting. And Rosa Franklin did what? Rosalind Franklin did key to DNA structure via X-ray diffraction, but not the DNA fingerprinting. Here is the picture of Sir Alec Jeffrey. Now, what is the basis of DNA fingerprinting? First, RNA sequence. Second, variable number of tandem repeats, VNTRs. C, protein sequence of fourth single nucleotide. So it is what? It is variable number of tandem repeats, that is VNTRs. VNTRs are what? They are highly polymorphic DNA regions. Okay, there's a lot of variations individuals among the population. That's why create unique patterns for individuals. Part of satellite DNA. Okay, and this RNA sequences, DNA fingerprinting analyzes DNA or not RNA. Protein sequencing, DNA fingerprinting analyze DNA, not protein. And single nucleotide, this like doing what? It is doing DNA fingerprinting focuses on repeating DNA nucleotide, not individual nucleotide. So here this one thing. So what happened? This part of satellite DNA. This is not a part of satellite DNA. They are just saying that this VNTR are what? This is the part of the satellite DNA. This RNA sequence, protein sequence, and single nucleotide. These are the other options, and they have answer like why these are not the correct answer. Why DNA sequence is not the correct answer? Because this is the DNA fingerprinting analysis. DNA, not RNA. And we know that DNA fingerprinting only analyze DNA, not protein. And if we talk about single nucleotide, so this DNA fingerprinting focuses on what? On repeating DNA sequencing, not what? Not a single nucleotide. So that's why the other three part was not the answer, whereas this VNTR, which is a part of satellite DNA, is the answer. So these are the short tandem repeats which we have discussed when we were discussing that part. Now, NCERT question number 3, that what technique uses restriction enzyme and DNA analysis to create unique identification pattern and is commonly used in forensic science. So this is very easy. This is for today's topic. Either it is DNA fingerprinting, is it karyotyping, it is gene sequencing, or it is chromosome banding. So it is DNA fingerprinting. What is DNA fingerprinting? It is based on unique DNA pattern of individual, uses restriction enzyme to cut DNA. So here are the different steps of DNA fingerprinting. What is the step? First, they have extracted the DNA sample. Okay. Then they have used restriction enzyme to cut the DNA fragments. Then they run electrophoresis. Long DNA fragments will be like a verb. Short will come beneath. Then they transfer it to membrane. So this is what the southern blotting we were talking about. What they did, they took this gel, they this transferred it to the membrane. It is made of nylon or nitrocellulose, and now we have incubated this. Membrane was already incubated with what? VNTR probes. So after that, if you will see on like X-ray light autoradiography, you will see the DNA fingerprinting. So this is the steps of DNA fingerprinting. Now let's see. Important in what? In forensic as well as paternity testing can identify individuals with high accuracy, because we know this will be same in only identical twins, not any other people. Now if you talk about karyotyping, so it is this analyze the chromosome number and appearance. It detects the genetic abnormalities, not individual identity. If we talk about gene sequencing, it determines the precise DNA sequence, providing detailed genetic information but rarely used for forensic ID. Then chromosome banding stains chromosome to visualize bands, used for identifying chromosome abnormalities, not individual identity. Let's see next question. Next question is, what type of membrane is used in southern blotting? Is it cellulose acetate, glass, plastic, or nitrocellulose or nylon? So it is nitrocellulose or nylon. Nitrocellulose, it is historically nitrocellulose membrane were most commonly used in southern blotting. This ID because of their ability to bind DNA effectively. This is used because of their ability to bind to DNA effectively. Of if we talk about nylon, it is more recent advancement have favored the use of nylon membranes. They often have a higher binding capacity than the nitrocellulose and can even withstand harsher conditions. If we talk about the other option, that is cellulose acetate is a membrane material. It is not typically used in southern blotting because it don't have very high binding capacity for the DNA. Then glass we are talking about is not used as a membrane in southern blotting. It does not have any necessary properties to bind to DNA. If we talk about plastic is also not suitable for southern blotting. Now next question is, why are VNTR useful for DNA fingerprinting? Either they code for essential proteins, they are identical in all humans, they show high variation between individuals, or they are present only in criminals. So we know the answer. The answer is they show high variation between the individuals. VNTRs are what? These are the short DNA sequence that are repeated variable number of times in different individuals. Pattern of VNTR repeats is highly unique to each person. This makes them ideal for identifying individuals through DNA fingerprinting. And these are the short tandem repeats. Then VNTRs are not typically in non-coding regions. VNTRs are highly variable, enabling individual identification only. That's why they're not identical in all human. VNTRs are present in everyone's DNA. It is not specific to criminals. Now, what is the purpose of what? What is the purpose of the probe in DNA fingerprinting? To cut DNA, to detect complementary sequence, to separate DNA fragments, or to extract DNA. So we know that the probe is used to detect the complementary sequences. A DNA probe is a short single-stranded piece of DNA that that is designed to bind specifically to the complementary sequence within a large DNA molecule. It enables visualization of specific DNA fragments, and we can see like how we can use this DNA probes. This was a DNA sample. Okay, we have isolated the DNA from body fluid sample. Then we denatured it. Okay, this is our gene of interest. So what we will do, we will add now DNA probes, because what DNA probes are what? These are the short single-stranded piece of DNA which is complementary to this gene of interest. And in this is radio labeled. So now what will happen? This probe will go and bind to its complementary region, and this is how we can visualize it and identify it. And this restriction enzyme is used to cut the DNA, not probes. To separate DNA fragments, we use gel electrophoresis, not probes fragment. To extract DNA, so DNA extraction is a separate process that involves isolating DNA from sample. So yeah. Next question: which of these is not an application of DNA fingerprinting? Is it is it what? Is it paternity testing? Is it criminal identification? Is it blood type determination, or is it immigration verification? So it is blood type determination. DNA fingerprinting is used for forensic science, parentage testing, immigration verification, but not for blood typing. Now, next question came. Next question is like, what is satellite DNA? Okay, so satellite DNA is either DNA found in satellites, DNA that forms separate peak during density gradient centrifugation, DNA found only in space, or DNA that codes for important proteins. So we have studied this in like first or second slide, third or fourth slide. That DNA that forms separate peak during density gradient centrifugation. Genomic DNA can be separated using density gradient centrifugation. Repeated sequence have different densities than the main DNA. These differences cause them to separate into distinct satellite peaks. So this was the picture. So when this, you know, density gradient centrifugation has been done for the whole genome, what happened? We have seen the smaller peak, okay, and that peak was like satellite band, satellite DNA. This DNA found in satellite, this is a misleading analogy. The term satellite refers to the separate peak which is observed in centrifugation, not actual satellites in space. DNA found only in space. Satellite DNA is found in the genomes of most eukaryotic organisms, not in space, not just in space. DNA that codes for important protein, we know that this satellite DNA is primarily non-coding, meaning it is not going to translate into protein. Now, next question: in DNA fingerprinting, what is the final step? Is it DNA isolation, gel electrophoresis, autoradiography, or restriction digestion? So we know that it is autoradiography. Why? Because in autoradiography, it is the final visualization step. Uses X-ray film, show dark band where probe has bound. So this is the what? This is the autoradiography in which they have used X-ray. Whereas DNA isolation, it is the initial step in the DNA fingerprinting, where actually what happened, where the DNA is extracted from the sample. Then gel electrophoresis, this step separates DNA fragments based on their size by running through them across gel. Then we will talk about restriction and digestion. This is like what? This is a step in which this DNA is cut into different fragments using restriction endonucleases. Next and last question is, which statement about VNTR is false? Options are: they are unique to each individual, they code for essential proteins, they are highly polymorphic, or they are part of repetitive DNA. So, three among are true, one is false. What is that? That is second one, that it is essential for Protein coding. We know that VNTRs are non-coding DNA, does not code for any protein. They are unique to each individual, means different individuals have different number of repeat at each VNTR locus, making them highly individualistic. The example we can see, like, one it is like 4 repeats only, for other it is 6 repeats. They're highly polymorphic. They show a lot of variation between individual due to varying number of repeats. They are part of repetitive DNA. VNTR are considered as a like type of repetitive DNA, meaning their sequence is repeated many times within the genome. What are today's keynotes? That DNA fingerprinting was first developed in 1984 by Alec Jeffrey, can identify identical twins, only need 100 cells for DNA analysis, 99.9% accurate in identification, used to solve first criminal case in 1986, can determine paternity with 99.99% accuracy, DNA remains stable long after death, no two people have identical DNA except identical twins. And when we are saying that we can identify identical twins means because they both have going to be same DNA fingerprinting. So these are the keynotes, and now the interesting questions. First is, why can't DNA fingerprinting differentiate between identical twins despite being able to uniquely identify all other individuals? Then a crime scene investigator finds a single strand of hair at a murder scene. How could DNA fingerprinting help solve this case? And what essential step must be checked about the hair sample? Hint, think about whether all hair sample contain DNA or not. Then, if VNTRs don't code for proteins, why are they so valuable in DNA fingerprinting? Fourth, in a paternity case, if a child DNA fingerprint pattern shows 10 bands, how many of these bands should theoretically match with biological father's DNA pattern? Think about the inheritance pattern. Then, why is it crucial to use radioactively labeled probes in DNA fingerprinting? What would happen if the probes weren't labeled? So these are the interesting question. Thank you. So here's the DPP. First question is, what is the primary basis of DNA fingerprinting? We know that it is variable number of tandem repeats. Then, which step in DNA fingerprinting involves transferring DNA fragment to a synthetic membrane, that is southern blotting, in which what we do, we transfer the gel from this DNA from gel to synthetic membrane, that is nitrocellulose and nylon membrane. In DNA fingerprinting, why do VNTRs produce distinct band patterns for individuals? Why? Because their copy number varies between chromosomes and individual. Which application is not associated with DNA fingerprinting? It is determining blood group types, whereas this forensic identification, paternity testing, and studying genetic diversity in population can be done. Then, what is the significance of using PCR in modern DNA fingerprinting? What it can do? It can, like, it amplifies DNA, enabling analysis from a single cell. Okay, so these are the like today's DPP. Let's save it. Submit. Yes. So here you can see all options are correct, all answers are correct. So yeah, it's all for today class. Thank you.